Today I'm going to talk about something that basically different speakers have already touched upon in their own talk, like Zhigang, like Kershaw yesterday. It's basically a class of nonlinear embedding methods that are very popular in applied field. So the goal of those algorithms again is sort of classical, is just for dimension reduction to have a lower dimensional representation of high dimensional data. We hope that the underlying structure is well preserved in this lower dimensional. To just locate this family of embedding methods along the history, we can see a timeline of a group of algorithms. At the beginning of 20th century, we have principal component analysis. And then at the towards the end of 20th century, we have first a large variety of linear embedding methods like kernel PCA, Gaussian eigenmap, local linear embedding, etc. And the family of algorithm that we're going to focus today is basically starts from this 2008 publication in Journal of Machine Learning Research called t-SNE, this algorithm proposed by Van Martin and Hinton. And after that, there are another several algorithms, but more or less they belong to the same family called neighbor embedding algorithms. They are modeled after this t-SNE algorithm. Another important representative algorithm that are equally popular is this UMAP. As many speakers already show some of their results using this visualization tool. So we're going to focus on about that. And just for the background, this algorithms represented by t-SNE UMAP has nowadays a dominating role in biological research. And you can see in the top scientific journals, Nature and Nature Biotechnology, there are papers dedicated to this group of algorithms towards computational biology or biologists talking about how to artistically use those algorithms for biological analysis, especially for single cell transcriptomic data. And you can see from 2018 and up to last year, there are a series of papers just talking about those algorithms alone. To get a sense of why the algorithms suddenly become popular in applied field, we can look at this simple example. Here, the goal is just to visualize a large data set containing 6,000 images of handwritten digits. So each data point or each sample is just this image of digits, handwritten digits. And basically, we can consider this data set for each data point is a long vector, which is a vectorized version of this grayscale image based on their pixel. And then, of course, behind each image, there is a true label telling us which digit it is. And there are 10 different digits. So we consider there are ground truths, like 10 different classes, clusters. And here we apply 6 different nonlinear or just linear embedding algorithms to find a 2-dimensional representation of this data set. We do not use label at all to obtain the embedding, and then we just color those labels to see how well they separate those different images. And we can see the classical algorithm PCA, although it only took 2 seconds, but basically it does not separate different class of images very well. And there are 3 alternative nonlinear methods. The performance compared with this t-SNE UMAP, they're considerably worse. Not only that, the algorithm is very fast. As you can see here, UMAP only took 16 seconds compared with, let's say, ISOMAP, which need 2 more minutes. Okay. So although the algorithm shows very pleasant performance in separating or identifying cluster patterns in the data set, when the algorithms are proposed, they are just the iterative algorithms with some heuristics of objective. There is no clear reason why this algorithm should perform largely better than the existing nonlinear embedding algorithm. So one motivation for our study is to understand why they work that well. Another motivation is that if you ever use those algorithms, you will see that there are actually quite a few tuning parameters associated with this. And it turns out that the algorithm performance are pretty sensitive to the choice of those hyperparameters. Here you can see the two examples. We just apply UMAP to the same data set, but with different tuning parameters. We just change one tuning parameter, consider two different values. And you can see the resulting embedding look very different. In one case, it look like a single cluster. In the other case, it seems to be more clusters showing. And the situation is even worse for t-SNE. As we will explain shortly, t-SNE is a randomized algorithm. Basically, if you apply algorithm to the same data set under the same parameter, hyperparameter, you may still have very different outcome. So in this particular example, you can see one case, you have seemingly 5 clusters, and in other case, you have seemingly 4 clusters. So the number of clusters obtained from the algorithm may even be different, which can be practically quite disturbing. So better understanding of those algorithms help us to guide us how to use those algorithms in a principled way. So this is a quick outline. We're gonna focus on t-SNE, basically the first algorithm in this family, to show you how it is designed, and then we're gonna discuss this algorithm more depth. So the algorithm is pretty straightforward. The input is just a bunch of high-dimensional vectors xi. The output in this particular case is just for visualization purpose. So the output will be 2-dimensional vectors. But the algorithm, as I described, basically does not require this to be 2D embedding. It could be any finite dimensional embedding. So the algorithm's idea is to construct the 2 similarity matrices, one from the original input data, xi's, the other associated with the output low-dimensional embedding. So the first similarity matrix P is essentially a normalized version of the pairwise Gaussian kernel matrix. So you start from the original Euclidean observations, you construct Gaussian kernel with some bandwidth, and you normalize that K matrix into this P. The normalization is essentially to make sure the row sums, column sums are all equal. And another operation is to make sure the sum of all the intrinsic matrix is 1. That's just to make sure the similarity matrix P is invariant to the scale of your input data. And now for the low-dimensional embedding, which is the goal of this optimization, we can also define a similarity matrix, K prime. And here, the author did not propose to use Gaussian kernel for this output embedding. They use this particular kernel function, is, if you recognize it, is the density for the Cauchy distribution, the heavy tail distribution, or student T distribution of degree of freedom 1. And it turns out that the choice of heavy tail distribution here is essential to the success of the algorithm. And to highlight that, the authors named their algorithm t-SNE. So the T in front is to highlight that here they use the T kernel. So a natural option, of course, would be just to use Gaussian kernel in both cases. But it turns out that after trying different things, they find that this is very good. And now after K prime, you can also normalize the interest so that sum of all entries equal to 1. That gives you Q. And the objective of t-SNE algorithm is to minimize the discrepancy between these two similarity matrices, solve for the low dimension YIs. Of course, this optimization is pretty complicated. You don't even have a closed form solution, or don't even know this is convex, or don't know whether there is a global minimum. But we can compute this through a gradient descent algorithm practically. So since the objective is known, then you can just numerically compute the derivative for the objective with respect to each yi. And for given initial value, you just update the embeddings gradually. However, the t-SNE algorithm, in the paper, they made important modifications of based on this gradient descent framework. So the algorithm consists of three major steps. The first step is initialization. The paper proposed to use this local initialization, where you generate the initial embedding of your data from a tiny neighborhood of this two-dimensional plane. Small values, but random. Second step, they call it early exaggeration stage. So in this stage, instead of working with original gradient descent updating equation, they just look at this updating equation where you locate the P matrix similarity construct from input data, and you multiply each entry of this P by a large constant, alpha. So this is the only place that are different from the original gradient descent algorithm. And in the standard software implementation, this alpha is just set as some constant. In the R implementation, it's just 12. And using this modified updating equation, the algorithm will run it for some k0 times. And again, in R, this is a 250 times equation. And afterwards, you go back to the original classical gradient descent to run it more times. So these two steps remains quite mysterious for a long time. And in the paper, the author explicitly stated that this early exaggeration mixed with local initialization was kind of an empirical strategy that they literally explored around different combinations of things to improve the performance of algorithm. And they find that actually these two combined together is really good. And they cannot find something similar. And although we do not have an intuition of why it should work or why this is the case. So for the next few slides, we're going to focus on embedding of cluster data. Basically, we're considering in the input data set, there are some cluster patterns already. It could be a strong signal case where you know that the clusters are already separated in the high dimensional space, but it's because of the dimensionality you cannot visualize. Or it could be a messy case where you have overlap between clusters. We're going to start from some empirical observations of t-SNE performance, focusing on its early exaggeration stage. We generate Gaussian mixture data consisting of 6 clusters. It's the 6 different colors. And we look at the embedding of t-SNE produced by its iterative algorithm. So this is the initial initialization, random small values. And then we run this t-SNE early exaggeration for a few times. And as you can see, if you move on to more iterations, you see you have 2 phenomena. First of all, you see convergence of the points belonging to the same Gaussian mixture class to eventually to a single singleton collapse into a single point. And second, the location of those clusters are seemingly random. Why is that? We can just generate another random initialization and run the same procedure. And you can see from the second time initialization, the location of the limiting dots completely changed. So we want to figure out that, first of all, whether this two findings are some principal behavior for the algorithm, or it's just for this simple case. Okay, so first of all, this basically cluster have already been identified or created throughout the early exaggeration stage. And the location of the cluster centers seems to be random. So we did a analysis of this iterative algorithm. What we find, the key observation is follow. So we find that this early exaggeration stage, this iteration, so we can rewrite this updating equation into a matrix form. And we find that the low dimensional embedding produced by t-SNE, this early exaggeration at the case step, is in fact approximately a power iteration on the initial value. And this multiplier matrix is identity minus some scalar. Alpha is, remember, alpha is the early exaggeration. H is the step size of your updating equation. And this is a Laplacian operator of your similarity matrix of the input data. Okay, so you can see this is a very similar to kind of a Laplacian effect, given that power iteration essentially look for the top eigenvector of that matrix. So once we know that, we have this approximation because power iteration is well understood and Laplacian operator also well understood. In that case, we can analyze the asymptotic behavior of early exaggeration by just through this approximation. However, you may wonder that how this is related to their step 1 and 2, right? It turns out that two computational strategy is essential to make this approximation true. So we write the original updating equation with the early exaggeration parameter. We can recognize a form that is already very similar to a power iteration, except for that within this Laplacian operator, this is not a constant matrix. This is a matrix that also updates itself through iteration. However, the variable parts of this matrix Q and Yi, Yj, their role are reduced under this first and second step. You can see if this alpha, the early exaggeration is larger than a constant, then in the numerator, the effect of this Qij will be dominated by that. And when you start from very small values, the first steps, the embedding are very small, then the denominator effectively becomes 1. So in that case, if you have early exaggeration plus local initialization, effectively reducing this S matrix into this Cij times alpha alone. That gives you this approximation form. Okay, with that, we can just analyze the limiting behavior for this early exaggeration, given that this is just a Laplacian power iteration. We know that in that case, the limit of the early exaggeration essentially is the null space of that graph Laplacian for the input similarity matrix. As a result, when the data are well clustered, meaning that the high dimensional data are already well separated in the original space, then we can show that the above two findings actually holds in the asymptotic sense. That is, you guarantee to converge to a few singletons corresponding to the cluster identity. And also the location of those dots are exact functions of the initial value, y0. So basically that's a random quantity. And with that, we can also interpret why the low dimensional embeddings produced by t-SNE for a real data set are similarly random in terms of their cluster layout. And as you can see in this example, these two clusters are nearby each other. If you write again, they are away from each other. So basically, the t-SNE embedding of clusters, the location of their clusters are seemingly non-informative. It's just a way to lay out different clusters. By the way, if you have any questions, since we don't have time afterwards, so if you have any questions, just feel free to interrupt. Happy to. Okay, so up to now, we're talking about the high signal to noise ratio case, right? The original data are already well separated. In that case, we will have that kind of nice behavior. But for a real data set, when the clusters are really noisy, it turns out that there are some different behavior, qualitatively different behavior. So again, back to this handwritten digits example, you can see after 50 times of the iterations in this early exaggeration, we already see separation of clusters at the classical case. However, if we keep running this algorithm for more times, instead of finding that they converge to those singularities, actually the embedding become more and more non-informative. And this is because when you have noise, you really do not have a nice null space for the Laplacian. And just using the same argument, we can show that if your data are really noisy in that case, the limit of this early exaggeration now become very non-informative. We call it overshooting. Because we can see that actually early in the stage, there are already something that are informative. By running it too many times, you actually lose that information. And another piece of result we can show is that in that noisy case, actually, even though the limit of the algorithm, the null space of the graph Laplacian is non-informative, but the path of this iterative algorithm itself is informative. Is that if you actually stop early along this iteration path, you can effectively capture the cluster pattern again, just as shown in this example. And the key is to establish this relationship through a gradient flow analysis, where we just analyze this iterative algorithm by sending the step size very small. We can find a continuous limit of this iterative process. And as a result, we can establish this approximate expression where the low dimensional embedding produced by t-SNE is approximately a weighted average of the eigenvectors of that graph Laplacian. And the weight is proportional to an exponential decay, where k is the steps, is the number of iterations, and lambda are the eigenvalues of the graph Laplacian. As you can see, for any contribution from the factors whose eigenvalues are non-zero, as k goes to infinity, its contribution gonna be zero. So the limit, again, this conforms to the fact that the limit of this procedure will be something only in the null space where lambda is exactly zero. However,
the large alpha there.
So
you still have the large alpha.
You mean this one?
Yeah, alpha.
Alpha, you still have a large alpha, like.
So we're still in the first part inflation.
Still in the early exaggeration state. This is a behavior for that stage alone. And what happens if you stop early? When you stop early, this is effectively a weighted average of the leading eigenvectors. And the weights depend on the importance. And in that case, we can see if we stop early, we effectively achieve a soft regularization of soft thresholding of the top eigenvectors based on their corresponding weight. That's just how it ignores the data. Okay, so through this connection between early exaggeration and Laplacian power iteration, we see that in this procedure, clusters are created in visualization. That's why these two steps combined together will very fast, quickly find the cluster patterns. As a result, it speed up the convergence of the embedding. And by early stopping, we can denoise the data to see the cluster. and also explains this random cluster positions. And now I want to compare this t-SNE with many other algorithms along this within this family. So t-SNE and another algorithm called large V's, they have the same procedure, basically early exaggeration plus the initial random initialization. And in both cases, as we can show, see from the previous slides, it's kind of soft threshold spectral initialization. It's just the weighted average of the. Yeah.
Question about the previous slide. If we stop early at some point, and how would the formula change if I stop a little bit earlier or a little bit later? Like what would be the
Right. So here, in theory, we can establish that for a given level of noise, signal to noise thread, there exists an optimal range of K, so that this embedding will converge to basically converge to something that separate the cluster as well. But in practice, we actually just know that we just know that this optimal stopping time is a function of log n, and its number of data points. But the constant in front of log n is depending on the actual signal to noise ratio. Alternatively, another class of algorithm, including UMAP, actually is starts from spectral embedding. So the initialization is spectral embedding. That be PCA embedding, could be MDS embedding, or directly Laplacian eigenmap. In the case of Laplacian eigenmap, there is a very interesting connection between the two. In the first one is weighted average of all the eigenvectors ranked by their corresponding eigenvalue importance. However, in the Laplacian eigenmap is kind of hard spectral initialization, where you just given a K and then we just use the top K eigenvector for the. So t-SNE basically get around of the difficulty of choosing the latent dimension K by transforming into the number of iterations for your early exaggeration. Okay, now if we move on to the last stage of the embedding, back to this example and written digits. So after 50 times of iteration where we feel it's good for the early exaggeration, we just back to the original mating equation and run it more times. What we can see is that the location of the cluster centers are roughly preserved as the output of the early exaggeration. However, the separation of cluster is even more it's better. And this actually we find has something to do with the algorithm design itself. So just in order to empirically assess how well t-SNE or UMAP, those algorithms preserve the cluster patterns, we just look at this two-dimensional input data. So this is a Gaussian mixture of two clusters. We intentionally make them to have some overlap. And we apply t-SNE UMAP to see what the embedding look like, that type of thing. This is not even dimension reduction. Just want to see whether this map is continuous or something. You can see the t-SNE embedding and UMAP embedding in both cases intentionally separate the two clusters, creating a space in between. And in this example, we just put a point from the center of the original input data one cluster, move gradually to the center of the other cluster and track its embedding in the location of this embedding in that space. And you can see as we move continuously from one cluster to the other cluster, its corresponding embedding actually experiences a big jump from one cluster to the other. Basically, the algorithm literally does not allow any point to appear in the middle. So basically, there is artificial gap or vacuum created by the algorithm, so that you don't have anything in between. The consequence is that many points of order supposed to lie in between or sent inside the other clusters. Another drawback of this procedure is that it creates kind of a misunderstanding or artificial conception about the data, that you will feel that if you never be able to see the original data, you will feel that after visualization, you will feel that the data are already well separated in clusters. You have very strong signals. However, this is more or less an artifact of algorithms. Yeah, so from that empirical assessment, we can see as a function, those t-SNE embedding as a function from high dimensional space to low dimensional space is essentially discontinuous. There's a jump in the output space. And it has a practical consequences. So all of is so far the biggest genetic studies in US, and recently they published their flagship paper in top scientific journals. In one of the scientific results, they actually made a UMAP of all the populations under study based on their genetic profiles. And as a result, they can the UMAP produced these big clusters of populations. And immediately after this paper published, there are a lot of criticism from Twitter or X talking about the validity or reliability of this visualization, because seemingly this UMAP visualization exaggerates separation of different ethnic groups, and where people believe that there should be a gradual, continuous shift from one to the other, where we have a lot of admixtures. But in this case, you have very clear separation, clear cut, and people related this to the artifact of the algorithm. And mathematically, we can actually prove that in this stage, what happens is that suppose the clusters already created at the end of early exaggeration stage. The algorithm, iterative algorithm at this stage, effectively push those clusters away from each other toward the direction that are determined actually by the relative positions of those clusters. And exactly like the physics of the repulsive force. And like in this case, this thought at the next step will exactly be here. And the direction is proportional to the distance from the other cluster. And as a result, the clusters will just be proportionally pushed away from each other like an explosion. And this mechanism actually increases the space between the clusters, just push them away from other even further. And here I want to make a second comparison with, because our analysis is based on t-SNE alone. We want to compare with other algorithms in the group, in the family, to see how similar they are. So just for t-SNE, remember we have three components: random initialization, early exaggeration, gradient. For UMAP, it's Laplacian eigenmap plus gradient descent. And from previous discussion, we already see the connection between these two combined together versus the button. And now comes the gradient descent. So someone wrote a blog, basically compare the gradient terms for different members of the algorithm in this family. Just ignore the meaning of those parameters, just to compare the patterns. You can see the gradient for t-SNE look like this, and the gradient for a large V is another algorithm look like this, which is already very similar, just subtract and twist. And the gradient for UMAP is much more complicated. Although here A and B are just the tuning parameters specified by the users. If you fix A and B equal to 1, and just after some reparameterization, this can be reduced by this form. And now, although you cannot see the similarity between the two, but you can actually rewrite this in this form. Same thing. And now you can compare the similarity between these two. So what I'm trying to say is that all the three algorithms share very similar gradient term. And if you compare numerically, actually, they actually perform very, very similar. And the benefit of UMAP majorly is to provide this extra flexibility of tuning parameter AB so that you can twist your like some sense. Yeah, so similar observation have been found by computation biologists. They just through numerical experiment, they find that if your data, say, is manifold, then it seems like the performance of the t-SNE UMAP is just completely determined by how they initialize their algorithm. So the default initialization is Laplacian eigenmap for UMAP, whereas for t-SNE it's random initialization. And for t-SNE you have this, basically you broke the global structure, whereas for UMAP it seems better. And the author is trying to argue that this is just because of the choice of default initialization scheme of the algorithm rather than the algorithm itself. If you just switch the initialization, actually you can have a even slightly better initialization embedding for t-SNE. Whereas for UMAP, the random initialization is something the same. So for this last stage, the embedding stage of cluster data, we know that this algorithm by design exaggerates cluster separation. It's both good and bad, depending on what you need. If your goal is just to recognize those clusters, perfect. That's why people like this UMAP. However, if your goal is either to interpret the relative position, the relationship between those clusters, it might be dangerous. Or if you want to interpret your visualization as an honest isometry of your high dimensional data. Good. And this separation we notice is due to the intrinsic discontinuity of the embedding. And people find it likely with the global structures if you do not initialize the algorithm carefully. So at the last part, I want to talk about people, although the popularity of t-SNE UMAP was originally due to its very good performance in visualizing cluster patterns in high-dimensional data. But nowadays, people also use this as a tool to visualize the manifold. They will consider that maybe apply this to some data set with continuous structures. So we may visualize something like a trajectory or something more continuous. So the question here we want to ask is, first of all, what are the possible artifacts of those algorithms when it applied to the data set that contains smooth structures? Second, how to overcome those limitations. And in this connection, we actually collaborated with ambiguous group at UC Berkeley. Their group recent years, they're dedicated to this PCS framework, basically promoting the stability principle, I would say, in statistical procedures, and they published a book on this. And just to focus on our problem, here we looked at the real single-cell RNA-seq data set of blood cell development. So the experiment is we draw samples from human bone marrows, and we have about 3,000 cells. And those cells are actively undergoing development. So this is the cell lineage tree. And basically, we have cells at different stages of this development. And we have just their gene expression measures for about 3,000 genes. And now we apply t-SNE to this tabular data of cell by gene matrix, and we visualize those cells. In the ideal case, if we select the tuning parameter carefully, we will be able to visualize this tree structure and the colors corresponding to their ground truth. So basically, in this case, we know that the visualization can actually recover something of biological interest, whereas
Is this about differentiation of stem cell like cells?
yes, exactly, including from stem cell to red blood cell. Yes. And complexity is in fact the same thing as the bandwidth parameter for your P matrix, the Gaussian kernel. So perplexity is big, it means your bandwidth. So it controls the smoothness of your similarity matrix. And the conclusion here is that if your perplexity is too small, basically you will have a very fragmented visualization. However, it seems like the algorithm was able to recover this tree structure in some sense if you choose very large bandwidth. And now we look at some toy example back to the search case. So it seems like the choice of complexity determines how well you preserve the global structure. It's similar as you start with constructing nearest neighbor graph. If the graph is fully connected, then it's likely that for the global geometry. And in this case, we actually prove the simple result for t-SNE alone. Basically, if you know that the data are drawn from a one-dimensional manifold like this case, we can show that if the perplexity is not large enough, that actually there is an upper bound for that. Below this threshold, the low-dimensional embedding must contain continuity, discontinuity form. Basically, what we in fact prove is that when the perplexity is below something, the Lipschitz constant for the t-SNE as a map from high D to low D, that Lipschitz constant is going to blow up to infinity. That's so it turns out that because of the way this t-SNE global this optimization is designed, the minimizer in low complexity regime supposed to be fragmented. And another aspect is that remember t-SNE is a randomized algorithm. It turns out that if you're in the underspecified regime, if you run t-SNE twice, in addition to the fragmented nature, you also recognize that the low-dimensional embedding are highly unstable. Unstable in terms of its neighborhood structure. Let's see, in the first case, the red dots are here or far away from the other branch. In the other case, this purple dots are close to the other branch. So basically, it can subject to a lot of variations. Whereas if you're in the sweet spot, then the low-dimensional embedding are mostly preserved in terms of global structure, just subject to certain notation. So as a result, even if you don't know the underlying true structure, you can use this stability as a criteria to see whether you're in the regime where you have reliable visualizations. So as a consequence, we just design an algorithm based on stability because stability is something that you can measure. You can just regenerate t-SNE embedding many times, and then you just compute the stability of the embedding. And in that case, you will see that in the scenario where the algorithm seems to stably represent something as low dimensional would be correspond to some global true structure. So here are some just our examples. Yeah, so this mostly I want to share with you guys, and for the rest of the time, I'm happy to discuss more. Thank you.
So all you need is the t-SNE algorithm that should be.
Yeah, so for the last algorithm, it is the goal is actually twofold. One is to choose the good tuning parameter. In that case, the stability as a guide, right? Another thing that I just sort of skipped is that even if you're in the sweet spot, you can still compute the stability for each point. And in that case, you can interpret, let's say if so for a different tuning parameter, you can compute the stability, and then you can find somewhere that, you have high stability and gives you what to choose as. But this stability parameter can be both global and local. Basically, for each point, across different iterations how stable its neighborhood is. So we can have a point-wise score for your low dimensional embedding. So even if you just visualize a good embedding, you can still have an evaluation of which part is more reliable, which part is, the algorithm struggle with. And that result, in this case, as you can see, for a low dimensional embedding of complex data set, there are some parts that are basically more confident for the algorithm, and some parts are less confident. And in that case, we can find that this can be used to tell us something about the underlying biology.
Question just asked.
Did I understand correctly that if you have your perplexity large enough, you cannot push this pushing wave effect is less strong, or you not compared?
Yeah, so there are actually multiple factors drive this push away effects, including the perplexity. And in my case, in my understanding, I think that if you have if the underlying structure is continuous, basically it does not contain clusters, then the small perplexity will likely induce artificial clusters. The push away are just harmful, whereas the larger perplexity will prevent that. And I think the key insight here is that compared with the traditional dimension reduction tool like PCA, the success of this group of algorithms is that for cluster patterns, it completely gave up the goal of preserving distance between clusters. It's basically tear the space apart according to the cluster location, and then rearrange them into a grid.
But that's only for the two-dimensional t-SNE. If you do three-dimensional.
Anything actually, the analysis is independent from that. So even let's say if your data clusters are actually living in 5-dimensional space, if you do 5-dimensional t-SNE, still cut. It's arbitrary center.
You're saying one really needs to be very careful when you apply t-SNE.
Yes. So that's why some people even consider it not as a dimension reduction tool, rather as a cluster tool. It's basically assigning labels to data rather than consider it as embedding.
The same thing applied to the UMAP.
UMAP is better. UMAP, it's a spectral initialization. So it starts from PCA and updates from that. So in that case, the location of centers can have meaning.
Well, maybe this is stupid, but it's quite interesting that you have the quotient distribution of the and low dimension. So if you do higher dimension t-SNE, would you use higher and larger t's?
I have no idea. Yeah, so so far I do not have a clear intuition of why heavy tail distribution intuitively why it matters. But from the derivation, we can see that all those phenomena like tearing apart the space and the separation of major clusters, those are all due to this t distribution, where in the denominator you have a constant plus the Euclidean distance, so that basically if your Euclidean distance goes to 0, this function effectively goes to a constant, not 0. So it's not about the t distribution itself, basically just the shape. It could be a plus b times Euclidean to the 6th power over 1 over that.
I'm thinking of T2, T3, and so on, because they converge to a Gaussian. So
Yeah. I would say if it has to be heavy. If you T close to Gaussian, then you lose the.
Yeah, if you equal the dimension, then you just want to reproduce the clusters.
Yes.
Yeah,
So
very
it is
nice.
a distortion, but whether helpful or harmful depends on what you need.
It's a very nice talk, Jordan. So.
Thank you.